So I spend most of my time trying to figure out how that kind of effect happened. How is it that if we modulate social circumstances, we end up with changes in the activity of the human genome or the macaque genome or the mouse genome? And in general, we understand, at least at a conceptual level, pretty well how this happens. In large part, these dynamics are driven by social conditions' ability to change the way our central nervous systems work, to essentially influence how we're computing the, if you will, sort of the basic safety and generativity of our environment versus its sense of unpredictability, threat, perhaps outright danger. That change in how our minds and brains are making sense of the social world plays a central role in these gene expression dynamics. And one way we got a hint of this early on was by looking at the nature of the socio-environmental risk factors that did tend to have substantial associations with gene expression. There's, as you might imagine, lots of socio-environmental factors, emotional states, all kinds of things that turn out not to have much of a fingerprint on, let's say, our white blood cell transcriptome. So going through and finding what is the nature of the socio-environmental factors that do seem to have some kind of claim on this system's function, and saying how are they different from the ones that don't, was a productive thing to do. One of the things that emerged most saliently is the observation that subjective states, that they often loom as large or sometimes even larger than objective socio-environmental conditions in influencing the genome. So in this case, we were talking about loneliness. Loneliness is a subjective state. You can be lonely in a room full of people, and you can be connected with people and humanity while walking completely in isolation through the Arctic. You can have a big disconnect between what you might call the count of the number of people around, the number of people you interact with on a daily basis, and your sense of being alone versus connected to a cultural group, a family, a whole sort of race of human beings. And when you compare the number of genes that associate an activity level with subjective states, that's often larger than the number that associates with objective states. And interestingly, in this case, so loneliness tracks about 200 genes in significant differences in gene expression. In this particular study, there were maybe about 1/3 as many that tracked objective loneliness. And what was most surprising is that they weren't the same genes. There's a particular block of genes that was tracking subjective states and a very different group of genes that was tracking objective social isolation, or just the number of people you come in contact with on a regular basis. So these kinds of, if you will, subjective interpretations of external social conditions seem to be major drivers of these kinds of dynamics. And it's fairly well understood after that, if you're experiencing the world as safe and generative versus threatening and unpredictable, how you could easily imagine how differences in consequent neurobiological threat responses get disseminated out into the rest of the body through pathways such as the sympathetic nervous system or the hypothalamic pituitary adrenal axis. And they deliver neuroeffector molecules. You can think of these as biochemical representations of our perception of the social and cultural environments that we inhabit. And these interact with receptors on the surface of cells. This is, fairly well understood molecular and cellular biology, and that triggers these intracellular signal transduction cascades that result in the activation of transcription factors, the proximal protein regulators that control the activity, the transcription rates for genes in a highly selective pattern based upon millions of years of evolution that has essentially mapped different receptors via different transcription factors onto particular blocks of genes. So we know in general that is, if you will, kind of a schematic diagram of how conceptually this can happen. And what we spend most of our time trying to do is answer a few relatively simple questions about that abstract schema there. One being, which particular genes are actually sensitive to social conditions?